Saurav, can you hear me?
Yes, sir, I can hear you.
Hello. Okay, so we are supposed to do with the auxin. Now, as I introduced in the last class, that auxin is basically it is a phytohormone which is responsible for regulation of growth, and it has number of functions. So first of all, this particular auxin has its name derived from auxine. Auxine means it's a growth factor. So auxin basically is isolated from human urine. Is isolated from human urine. Right? So it has been isolated from human urine, and along with that, if we see that this particular hormone, auxin, it is also of three types, basically on the basis of chemical nature. On the basis of chemical nature, auxin can be divided into three types. One is auxin A, and then auxin B, and another one is heteroauxin. Heteroauxin. There are three different types of auxin. One is auxin A, auxin B, another one is heteroauxin. And along with that, if we talk about the different types of auxin on the basis of their availability, that means whether it is naturally made auxin, or it is synthetically made auxin. That means natural auxin, this is one, and another one is synthetic auxin. Now, for natural auxin, it is naphthalene acetic acid. Yes, this one will be indole acetic acid, IAE, indole acetic acid. Then naphthalene acetic acid, NAA. Then indole propionic acid, IPA. So these are different types of acids like naphthalene acetic acid, indole propionic acid, and many others like 2,4-D, 2,4-dichlorophenoxyacetic acid. Then I can write 2,4,5-T. That is 2,4,5-trichlorophenoxyacetic acid. So these are the different types of auxin that is on the basis of natural and synthetic auxin. Clear?
Okay, sir.
So if we talk about the different types of auxin, then along with that, we can say that this auxin is basically a growth promoting factor. It is a growth promoter. or it is a growth promoting factor. And if we look into this growth promoting factor, it has number of functions. What are the different functions which are associated with auxin? First of all, we can see that it is responsible for cell enlargement, that is cell elongation, cell elongation, then cell division, which involve karyokinesis, cell elongation, cell division. This is another very important function of auxin. And then there is another function, that is parthenogenesis. Parthenogenesis. What is parthenogenesis? First, I will be discussing point by point, that is how cell elongation takes place, then cell division, parthenogenesis. There is also another function, that is, it is responsible for maintaining the luster of the plants. It maintains the luster of the plants. This hormone is also responsible for growth, that is, trophic movement and apical dominance. So there are a number of factors like cell elongation, cell division, parthenogenesis. It maintains the luster of the plant. It helps in trophic movement. It also helps in apical dominance. So there are a number of functions. Along with that, some of the other functions, like it prevents abscission, which is the function of abscisic acid. So it prevents. It prevents abscission of leaves. So this is one of the function. Then also it helps in sprouting in potatoes, prevents sprouting in potatoes. So these are some of the functions which are associated with auxin, and also helps in xylem differentiation and callus formation. It helps in xylem differentiation and callus formation. callus formation. This hormone also responsible for formation of female flower. So it promotes formation of female flower. This promotes formation of female flower. So these are the different functions which are associated with auxin. So let us begin with the cell elongation. Sort of you can understand up
Yes,
to
sir, I
here.
can understand.
And the new child, I don't know your name actually. It's written Ayu. Can you understand up to here what I taught have written? Hello. Hope you can hear me, right? So these are the things. Let us first discuss with the cell elongation. So if I talk about the cell elongation, can I just mm, yes. In case of cell elongation, if I take a diagram of a cell, that means this is the plant cell and this is the cell membrane. So this is a plant cell. Inside this plant cell, there are some structures which are present. There is a receptor. This is known as auxin binding receptor, ABP1. Auxin binding receptor is present. To this, a hormone auxin will come and establish its contact. This is auxin. Now, this is having the information. This is auxin. It will come and bind with this receptor. This is known as the auxin binding receptor, or you can write ABP1. And inside this, there are some like as soon as this receptor will come and bind, there will be formation of a activation of a protein. This is a signal protein, right? This signal protein gets activated. From inactive condition, it will get activated to form active receptor. This will be the active receptor, right? As soon as this receptor becomes active, they are going to enter into the DNA. Suppose this is the DNA which is having the promoter, I think P, and this part. So here you can see that this is a active receptor which will go and bind with the DNA, and it will be resulting in formation of some protein. That means first it will activate the genes. Which genes? Activate the genes of H plus ATP, ATPase. Activate the genes of H plus ATPase. Right? So after translation, the mRNA which is formed this is having the messenger RNA for H plus ATPase, and from here there are endoplasmic reticulum on which the proteins are synthesized on the ribosome. Like this. So here what will happen? This particular protein will come up over here. This is the H plus ATPase protein. And then after, we can see that this protein will go and bind with the cell. Yeah. This protein will go and bind with the cell membrane like this. As this will go and bind with the cell membrane, it is going to pump out H plus ion over here. And
Okay, sir.
yes, by breakdown of ATP to ADP. It will break down the ADP to ATP, and in this way, H plus ion will be pumped over here. Right? So if H plus ion concentration increases over here, what is going to happen? That is the pH over here in the cell wall is going to decrease. The pH of cell wall will decrease.
Decrease.
In presence of this decreased pH of the cell wall, the cellulose microfibril will start breaking. It will break. And as the cellulose microfibril it will break, there is another protein which is associated with over here that is known as expansin. Have you done with the molecular inheritance, Saurav?
No, sir.
So after this we will, or the principle of inheritance not done, right?
Yes, sir.
No, matlab, have you done the principle of inheritance?
नहीं सर अभी तक नहीं किया.
ठीक है. तो एक्सपेंसिन एक प्रोटीन है which is responsible for expansion of cell wall. जैसे जैसे सेल वॉल का एक्सपेंशन होगा, न्यू सेल्यूलोज मटेरियल, this is new cellulose material will get it will enter into the existing cell wall structure. New cell wall material will enter into the existing cell wall structure. जैसे जैसे enter करेगा new cell wall material into the cell wall structure, that will result in elongation of the cell. This will result in elongation of cell.
Okay.
And moreover, another very important property of auxin is that it is having that is a feature of polar transport. Polar transport. Now, what is this polar transport? That means if this is the cell, this is the first cell, and then this is another cell. Cell to cell transport takes place in case of auxin. And then there is another cell. So there are some transporter which are present at the base, and there are some transporter which are present at the here at the apex. Again, there is another transporter which is present over here, and then again another transporter. Suppose this is present over here. In this way, the transporters they are present. Now, this particular transporter, which is present at the base, this is known as indole acetic acid, that is efflux channel. That means they are responsible for effluxing the indole acetic acid. They are going to pump out the indole acetic acid. So these are known as auxin efflux carrier, which is also known as the pin protein. This is called auxin efflux carrier,
Okay.
or this is also known as the pin protein. हेलो, आप थोड़ा सा पूछिए. This is auxin influx carrier protein, that is OX1. So there are two different types of proteins. One is a OX1 protein, another is the PIN protein at the base of the cell only. Now, how this is responsible? That means as this phenomena operate over here, यहाँ पे क्या होगा? There is a channel which is responsible for pumping the H plus ion. That means जैसे यहाँ पे हुआ था H plus ion का pumping हुआ था, same way that is ATPase is going to, like, 1 minute. In the same manner, this is the ATPase. It is going to pump H plus ion over here. And on the other hand, this ATP, it will break ADP, ATP से ADP हो जाएगा और H plus ion का entry होगा. So जैसे जैसे H plus ion का entry होगा, so here we can see that H plus ion and indole acetic acid which is formed, पहले तो दो फॉर्म में रहता है इंडोल एसिडिक एसिड. पहले प्रोटोनेटेड फॉर्म में रहता है और उसके बाद वो टूट जाते हैं क्योंकि यहाँ पे जो pH है it is greater. That means pH is more and यहाँ पे जो pH है it is lesser. So in IAA IAAH, ये जो इंडोल एसिटिक एसिड है IAAH, it will break into IAA minus and H plus. That means there will be breakdown of IAA minus and 2H plus. So IAA minus will enter over here. Here H plus and IAA minus. So IAA minus and there will be formation of H plus. So in this way, this particular protein, which is known as the auxin efflux protein, you know that they are resulting in efflux of this auxin. ये जो auxin है ना, IAA वाला, it is being thrown outside. It is being made to undergo. It is being made to come out from the cell. जैसे जैसे सेल से वो निकल के आएगा, it will enter into the next cell IAA minus or H plus ion. It will also come along with it. And then there is another protein which is present at the base. So IA minus will enter, that is with the help of OX1 protein, the IA minus will enter along with the 2H plus. So 2H plus will also enter over here. You can understand this 2H plus is present. So 2H
Yes.
plus and I, yes, 2H plus and IAA minus is present. That means we are studying how the polar transport is taking place. And this IAA minus which is present, there are another transporter that is known as the PIN protein, through which the indole acetic acid will be transported to the next. That means it is the efflux, auxin efflux carrier will result in transport of indole acetic acid to the next. In this way, indole acetic. So in this way they will be transported, right?
Yes.
So after this polar transport and after this cell elongation, after this polar transport and cell elongation, we can also study how it is responsible. What is parthenogenesis? What is parthenocarpy? Parthenocarpy is a process. Parthenocarpy is a process in which the fruits, that means the ovary, get transformed into fruits without fertilization. That means, say this is ovary. Generally, what happens after fertilization, ovary changes into fruit, and the ovules changes into seed. Ovary changes into fruit, and the ovules they changes into seed, and this takes place with fertilization. But in case of parthenocarpy, the ovary transforms into fruit, but ovules do not change into seed. This one, right? So this happens in case of parthenocarpy. So in this case, we can see that the fruits are present, but these fruits are without seed. So here, this will result in formation of seedless fruit. This is known as seedless fruit, right? So this will be resulting in formation of seedless fruit.
Okay, sir.
This is one way, that is parthenocarpy is one process in which the ovary is getting converted into fruit and there is no formation of seed. This type of fruit are available throughout the entire, that means you can get this fruit throughout the season by application of indole acetic acid. Now coming to another concept, like for that is seed germination, you can say the trophic movement, which I'm explaining one by one. Mhm. Trophic movement, that means it is a type of movement where you can say that in this movement the seed they germinate like this, you know. Suppose here it is sunlight is present. In this case, the hormone auxin will be shifted in this part, and this hormone will also be shifted in this part. So what will happen over here? This will show growth more. That means this is the premium, and this portion represents the radicle. So this portion will show greater growth, more growth, and as it will grow, it will give rise to this type of structure. it will give rise to this type of structure. That means when the growth will take place at this region, they will grow up like this. Growth will occur in this manner, and gradually here in this way the growth will occur. And this is a region where the growth is occurring in this manner, because you know that this region the growth is more, and this region the growth is less. In this region, and this is the region where growth is, okay, 1 minute, it is less. This region the growth will be more, because auxin is present over here, and at this region the growth is going to be less. So in this condition growth will be more at this region, and on this region the growth will be less.
Yes, sir.
Yes. So in this case we can find that it will be showing a trophic movement, and due to which the shoot, there is formation of shoot that is upside upward. That means a plumule will grow upward, and the radicle will grow downward. Downward direction. That means this is why this is positively phototrophic, and this is negatively geotrophic. Right? So this is the reason, that means this is the explanation of the trophic movement in case of auxin, that is a function of auxin. And also, as I mentioned, that these are the functions of the auxin. So how is auxin related to abscisic acid? Since we know that the plant hormones are not specific with respect to their function, auxin is related to abscisic acid in the sense that it is just the converse. It is just the reverse of the function of the abscisic acid.
Okay, sir.
That is the function of, yes, function of auxin is, that is antagonistic to the function of gibberellin, abscisic acid. Antagonistic to the function of abscisic acid. So after this, let us move on to the gibberellin. That means gibberellin is the another hormone which is very important. And like in case of auxin, as we have seen that it is a plant growth promoter, gibberellin is also a plant growth promoter which is responsible for growth of the plant in in other way, like not like the same way the auxin do. So let us move on with the gibberellin. So this part is very clear to all of you.
Yes, sir. It's clear.
Yes. Okay. Coming to gibberellin. Now, this gibberellin was first discovered from a fungus that is known as Gibberella fujiculae. It belongs to Ascomycetes. It belongs to Ascomycetes, and it was discovered by Kurosawa. And one thing about the auxin, that auxin, as I mentioned before also, that in case of auxin, the precursor of the auxin is tryptophan. So I can write it over here. The precursor of auxin tryptophan. Just let me shape it. Tryptophan is the precursor of auxin, and for gibberellin, you know that it is acetate. It serves as the precursor of gibberellin. We know that gibberellin is of various type like GA1, GA2, GA3, like this. And GA3 has been first isolated. It has been first isolated. GA1, GA2, GA3, there are different types of gibberellin, and GA3 was first isolated. And moreover, where you are going to get the gibberellin, it is found to be associated with the germinating seeds, then cotyledons, then you can see the embryonic axis, yes, which will after germination it will give rise to the embryonic axis, yes, the germinating seeds, including the embryo which are present inside the seeds. That means they are found to be associated in the mature seeds. In the mature seeds, this is the germinating seed, this is cotyledon, and this is a mature seed. And moreover, if we look into the function of the gibberellin, before coming to the function of gibberellin, the formula is for normal formula for gibberellin C19H22 O6. So this is a general formula for GA3 which has been isolated first. Clear? After,
Yes.
after this, we are going to move on to the different functions which are associated with the gibberellin. First is breaking of seed dormancy. This is one. I will draw and explain these functions. Second is breaking off, you can say like removing or eradication of this is breaking of seed dormancy. This is eradication of genetic dwarfism. Eradication of the eradication of the genetic dwarfism. Third point, which it can be like it helps in malting promotes bolting. It helps in parthenocarpy. It helps in parthenocarpy. It also helps in senescence. Prevents senescence. This is another point. And yes, it also helps in, it promotes formation of male flower. Is breaking of the seed dormancy, eradication of the genetic dwarfism, promotes malting, promotes bolting, parthenocarpy, prevents senescence, promote formation of male flower. So some of the functions which are relative, and also another that it helps in formation of flower in long day plant. It helps in photoperiodism. Photoperiodic effect can be overcome. Photoperiodic effect, uh, if not sufficient, uh, can be overcome by application of gibberellin. So here we can study so many features like breaking of seed dormancy, eradication of genetic dwarfism, breaking of seed dormancy, eradication of genetic dwarfism, promotes malting. So what are these breaking of dormancy and all these things? Let us see. I will first is breaking of dormancy. In your NCRT, there is a diagram in the chapter that is flower, where you can find the structure of the fruit. I'm trying to find it out. Yes, leaf. In case of monocot, So you know that this is a barley seed.
Yes, sir.
Yes, and this barley grain. This is the endosperm. Cutilum. This one is a radicle. This is This is coleorhiza. This is plenium. And this is radicle. This is coleoptile. So what happens whenever the gibberellin comes? It will bind with the receptor over here. That means this part, you know, that this is known as alluron layer. Alluron layer. Alluron layer. This is endosperm. Both of them, they are 3N. So whenever the gibberellin, they will come over here. Alluron layer. And as soon as the gibberellin will come and bind with alluron layer, it will convert the starchy endosperm to glucose. Now this glucose will be readily uptaken by the this portion of the plant, that is the embryo. This is the embryo. As soon as they will be taking the glucose, there will be germination of the seed. So glucose will be uptaken and there will be germination of the seed. So यहाँ पे seed का germination होगा. That means it will break the dormancy. It is responsible for breaking the dormancy. So that point has been cleared over here. Breaking dormancy of seed. And this glucose will be, so first what will happen? The gibberellin GA3 will establish contact with the receptors present on the alleuron layer. That means GA3 receptor complex. As soon as this complex it is formed, it is going to stimulate the alleuron layer to secrete an enzyme that is known as alpha amylase. Alpha amylase. Now, this alpha amylase will be acting on the endosperm and convert them into glucose. And this glucose will be readily taken up by the embryo readily taken up by the embryo and will help in quick germination and reducing the period of dormancy.